Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture by Chemi Biology. In this video lecture we are going to talk about Coombs test. Now what is Coombs test? Coombs test is a kind of test in which we can identify the antigen antibody interaction inside the body of a patient. What we can find? We can identify the antigen antibody interaction. Now here the second questions arise in our mind, that what is antigen and antibody? Antigen is a kind of molecules in which can activate the antibody. A kind of molecules which can activate the antibody. It will be outside or it will be inside our body as well. Now the second question is arise in our mind, that what is antibody? Antibody is a kind of molecules that are present on the surface of B cells, a kind of proteins. This antibody will be a protein in nature. So a proteins molecule that is present on the surface of B cells or T cells, we can say. So they are present on the surface of B cell or T cells, which will be produced on that time when the antigen are present inside our body. When the antigen are present and when this antigen is harmful to us, so then this antibody will be activated and these proteins will be activated. So that's about the antigen antibody. So we can identify antigen antibody interaction, or we can identify agglutination. So due to Coombs test we can identify the agglutination. Now what does it mean? It means that we can see that whether the antigen antibody are attracting with each other or no. So once we can get the antigen antibody attraction, so similarly we can get the agglutination. So in the case of agglutination, the RBC will be settled down at the bottom of the tubes. We'll be discussing inshallah in this video that how it will occur. So we just see the RBCs that are settled down on the test tubes. So this is agglutination. The second thing is about, we can see the kind of anemia. We can define the kind of, or we can say type of anemia. So due to Coombs test we can identify that which kind of anemia this patient have. Which kind of anemia this patient, either the patient have hemolytic anemia, or we can say immunohemolytic anemia, or we can say macro cytic anemia. Fine. So these are the different kinds of anemia. So due to Coombs test we can simply identify the kind of anemia. Second thing is about that due to this Coombs test we can identify what? We can identify autoantibody. So now what is autoantibody? So before going to explain this Coombs test, you need to understand about the autoantibody concept. Let's suppose inside our body, let's suppose this is the RBC. Fine. And what actually happened, this RBC have a kind of special, we can say molecules, we can say these molecules, let's suppose we can say that they have what? They have the antigen. So due to this antigen, antibody will be produced. So this is what? This is antibody which are against these antigen which are present on the surface of RBC. So once the antibodies are activated against this RBC, or we can say against this antigen, so what will be happen? This RBC will be destroyed. This RBC will be destroyed. So once the RBC will destroy, so after that what actually happen? We can say that it will cause immuno-mediated destruction. So this immunomediated destruction will be occur due to this autoantibody. So this is what? This is actually autoantibody. So what? This is actually autoantibody. So let me once again tell you. So let's suppose we have RBC inside our body, and this RBC contain a specific kind of antigen. This antigen will be a kind of blood group antigen, we can say blood group antigen, or we can say that this RH antigen, we can also say. So this will be RH antigen, or this will be the blood group antigen. So now we can also say that once the antigens are present and when the antibodies are activated, so due to this antibody the RBC will be destroyed, or the RBC when destroyed then it will cause immuno-mediated destruction. So when this immuno-mediated destruction are occur due to the presence of this autoimmune antibody, so then it will cause eventually this will cause what? This will cause this will cause anemia. So that's all about the autoantibody. Fine. I hope you understand about the autoantibody concept. Now let's talk about the Coombs test. Coombs test are actually, there are two types of the Coombs test. So let me write here two types. The first steps we call that direct, and the second we call that indirect. Direct Coombs test, indirect Coombs test. Now one important thing that I forget to mention, that about the Coombs reagents. Before going to explain this direct indirect Coombs test, first of all you need to understand about the Coombs reagents. Now what is Coombs reagents? First of all let me talk about the Coombs reagent. Now what is Coombs reagent? What is Coombs reagent and how we can prepare it? Now we can prepare it. That's, let's suppose first of all we take the blood sample. Let's suppose we take the blood sample from the patient. Let's suppose this is a patient. Let's suppose this is a kind of a patient. So let's suppose this is a patient, and inside this patient what we have? We have so let's suppose we have a patient, and this inside this patient these are the antibodies which are activated. These are the antibodies, and let's suppose this is a patient. So what actually we do? We simply take the blood sample from this patient. So once we take the blood sample, so then we centrifugate it. Centrifugate. After the centrifugation, what actually have we just separated the serum. So once we get the serum, so then we loaded this serum to an animal. So let's suppose this is an animal. Let me draw with a green marker. Let's suppose this is an animal. Any kind of animal, you can say a cat. So let's suppose this is a cat. Although my drawing is not good, but still I will try my best to clear your concept. Let's suppose this is a cat. Although this is not a cat, but let's suppose in this case this is a cat. And let's suppose we loaded the antibody inside this cat. So these are the antibody, or we can say these are the serum that we loaded here. Inside the serum, what actually these are? These are the antibody. Now what actually the cat, or what actually this animal do? This animal will also produce antibody against this antibody. So against the antibody that we are loaded here, they will also produce the antibody. So these are the antibody of what? These are the antibody of cat. These blue are the antibody of the cat, and these red are the antibody that we loaded inside the cat. So what actually we do? We just take this antibody, we separate this antibody. Right. So once we separate this antibody, and we simply call that what? We simply call that Coombs reagent. Now this is called Coombs reagent. Fine. So what is Coombs reagents? For making the Coombs reagent, first of all, we take the blood sample, we centrifugate it, and then we get the serum, and then we loaded this serum inside the animal, or let's suppose we can say inside the cat, and then these are the serum as we loaded, means these are the antibody. Due to this loaded serum, the cat will also produce antibody against this antibody. Now we simply take this antibody and we make a Coombs reagents. Coombs reagents is nothing but it is a kind of antibody which are produced against the antibody that we loaded inside the cat. Fine. Now let's talk about the direct and indirect Coombs test. First of all, let's talk about the direct Coombs test. Let me draw a line here. So here I will discuss the direct and here I will discuss the indirect. Now what is direct Coombs test? For direct Coombs test, due to direct Coombs test, what we can, now this is the direct Coombs test, and this is the indirect Coombs test. This direct Coombs test actually identify what this direct Coombs cell actually identify. Coombs test actually identify immunohemolytic anemia. So due to this direct Coombs test, we can identify the immunohemolytic anemia. We can see that whether this patient have suffering from immunohemolytic anemia or not. So how we can perform this test? We simply take the blood sample. Let's suppose this is the blood sample. Fine. This is the blood sample of, this is, let's suppose, the RBC blood sample that we take. And once we take the RBC blood sample, so then this RBC blood samples will contain what? These RBC blood samples, let's suppose, although we don't know, but let's suppose in this case they contain a kind of antigen, and due to this presence of antigen there will be an antibody. Although we don't know, but let's suppose they will contain what? They will contain the specific kinds of antibody on these RBCs. So first of all, we take the RBC sample from the patient, and let's suppose in this case these RBC have a kind of antigen, and due to this antigen the antibody will be present. So we take this RBC sample and we simply put it into a tube. Let's suppose this is a tube, and then we put it into a tube. So we simply put it into a tube. So let's suppose this is our RBCs, and we loaded this RBC in what? We loaded this RBC inside the tube, and this RBC will be also antibody, as we don't know, but let's suppose this contain antibody. And after that, when we want to check that whether this RBC have autoantibody or not, this is autoantibody as I discussed. We just want to check whether this RBC contain this autoantibody or no. For this purpose, what we actually do, we just loaded the Coombs reagents here. What we loaded? We loaded Coombs reagent. So due to this Coombs reagent, what actually happened? Let's suppose these greens one represents the Coombs reagents, means these greens ones are the antibody of what? These are the antibody of this Coombs reagents. Now due to the antibody, when the antibodies are attracted with the autoantibody, so then the agglutination process will be occur. So these RBCs will be settled down at the bottom. We call that what? We call that agglutination. Simply we can write it AGG. Agglutinations will be occur. So once we get the agglutination, so this test will be a positive, or we can say that yes, the patient have immunohemolytic anemia. Or when the patient don't have, or we can say when the patient have RBC, but these RBCs are not covered with what? These RBCs are not covered with autoantibody. So when the RBCs are not covered with autoantibody, and when we loaded the Coombs reagent, and when the agglutination are not formed, so it simply represents that this RBC are free, and this RBC don't have what? This RBC don't have autoantibody. So we can say this test will be negative, or we can say the patient don't have immunohemolytic anemia. So this is about the direct Coombs test. So due to direct Coombs test, what we can identify, we can simply identify the immunohemolytic anemia. Now let's talk about the indirect Coombs test. In this case, let's suppose we have the blood sample. Let's suppose in this case, let's suppose we have a woman. Let's suppose, and let's suppose this woman is what? This woman is get a pregnant. Inside this woman, what they have? They have a features. Let's suppose. Inside this woman, they have a features, or we can say this is a pregnant woman. So what will be happen actually? Let me draw a complete diagram. Although it is not the actual one, but it will just clear your concept. Let's suppose this is the mom, and inside the mom, what actually happened? They have a features. This mother have RH negative, and these features will be RH positive. So when the mother have RH negative and when the features have RH positive, so then before delivery or when delivery are happening, so then this RH negative and RH positive will be mixed to each other. Due to this mixing, this RH negative will produce what? They will produce antibody. We simply call that RH antibody. What we called it? We called it RH antibody. This RH antibody will be against to this features blood cells. So once these antibodies are produced to against these features red blood cells, so then the features will be die, or we can say the son, or we can say the babies which are present inside the mother will be die. Inside the pregnant woman will be die. So this is the first case. So before delivery, what actually the doctor will recommend it? The doctor will recommend it this kind of test, means indirect Coombs test. So due to this test, we can just simply detect that whether this patient have RH positive or not. So once we get that if this patient is RH positive, so then the doctor will give a kind of antibiotic, due to a kind of, not antibiotic, a kind of medicine. So due to this medicines, this antibodies will be not produced, and then delivery will be occur. Fine. So what actually happen? How we can perform this test? We can perform this test simply. We take the blood sample from the patient. Blood sample. Once we take the blood sample, then what we do, we centrifugate it. Centrifuge. Let me erase this one portion. We simply centrifuge it. So once we centrifuge it, so then we separate the serum portion. We separate the serum portion and we put this serum to a test tube. Let me draw here the test tube. So inside the serum, what we have inside the serum, we have the antibody, not the RBCs. So we have the antibody of this mother. Let's suppose we have the antibody. Let's suppose these are the antibody that we separated. So these are the antibody. Once we loaded the serum inside this test tube which contained the antibody, then after that what we loaded? We loaded the RBC. We loaded here RBC. So RBCs will be attracted here. So the RBC will be attracted here. So when the RBCs are attracted here, so after that what we loaded? Second we loaded here Coombs reagent. Coombs reagent. So due to this Coombs reagents, what we get? We get the antibody against this R2 antibody. So let me once again tell you. So then after that finally agglutination will be occur. So what actually this is Agglutination. Once again, tell me a little bit about. Due to indirect Coombs test, we can perform this indirect Coombs test for the parental screening of female Rh antibody. For the parental, let me write here. We can perform this indirect test for the parental screening of female Rh antibody. Fine. So we can perform this test for the parental screening of female Rh antibody. So for this purpose, we simply take the blood sample from a pregnant woman. So once we take the blood sample from a pregnant woman, then we centrifugate it and we take the sample. We simply take the serum, and once we take the serum, these serums will be contained. And although we don't know that this is contained R2 antibody or no, we don't know about that. But let's suppose this contained the R2 antibody, and we loaded this R2 antibody inside the test tube. So once we loaded this serum, we don't know about that whether there is antibody or no. R2 antibody or no. So once we loaded the serum here, so after that we loaded the RBC here. Next step is RBC here. So this is the first. This red one is the first one. This RBC, after that what we loaded? We loaded the RBC here. So once we loaded the RBC here, the reaction will be occur. But we don't know whether the reaction is occur or no. Or we can say whether the antibody are reacted with the RBC or no. For this detection, what actually we do? We simply, the third thing is what we are going to do. We are going to do Coombs reagent adding. We simply add the Coombs reagent. Once we add the Coombs reagent, so then agglutination will be occur. So once the agglutinations are occur, so it means that this patient have Rh positive. We can say this patient have Rh positive, and during delivery, these features will be die. During delivery, these features will be die. So what actually the doctor do? The doctor will provide a kind of histamine. Due to this histamine, or we can say a kind of drugs. Due to these drugs, the antibody will be not activated. So when the antibody are not activated, so the delivery will be easily done. So before delivery, we simply perform this test. Fine. So when the agglutination are not occur, so it means that autoantibody are not present in this case, and we don't give any kind of medicine to this pregnant one. We don't give any kind of medicine, and this is about the indirect Coombs test. Fine. I hope you understand about that, and that's all about the Coombs test. I hope you understand, and I hope fine. If you understand and if you know all about that, so then please hit the like button. If you have any kind of questions related with this video lecture, so write it into a comment. Inshallah, I will give you answer as soon as possible. So please like the video, share the video, and also subscribe the channel to get more interesting video lecture like that. Thank you so much.